Hello there. Welcome to the World of Sustenance, where your academic needs are met. We don't come again. We look on other parts of our lecture videos. This part now for our BCH people can also be used by other people too, other medical students or biological science students too. So this is an important topic because it is very essential. You know, in order to understand certain areas of our metabolism. So this topic today is on glycogenolysis. Glycogenolysis is an introductory lecture. You understand? So we'll keep doing more videos to actually buttress all of what we have been learning. So thank you very much. So from the normal procedure I follow, I must always digest. I must always break down the word to understand what we are talking about. So the topic here, glycogenolysis or glycogenolysis, any way you want to pronounce it, whether you spell it well. So the name glycogenolysis, glycogen, lysis. So glycogen is a polysaccharide, a storage form of glucose in animals, not just in animals, in microorganisms too. Lysis, breakdown. So everything we'll be looking at in this particular lecture or lecture series or lecture or topic is it has to do with the breakdown of glycogen. Very, very important, because glycogen is a storage form of glucose. You understand, in animals or in microorganisms too. So what I talk about and see how this glycogen take a breakdown. Name this topic now. So let's go right into it. This is a man or a person that is eating. They call it a fed state. You understand? It means that the person is getting energy source, glucose, fat, and what the point of interest here is glucose. You understand? After the person has eaten, or this guy has eaten, it would store the excess glucose as glycogen. But on the other hand, fasting state, or probably when the person is at rest, not eating after probably 4 to 5 hours or 4 to 6 hours, thereabouts, the person stored glucose, which is glycogen, would actually be needed so as to allow the blood glucose level be maintained. Blood glucose levels normally is around I think 4 to 5 millimolar. So very, very important. For the sake of this, there's a need for this person at rest or fasting. There's a need for that blood glucose to be maintained and also to help brain function, because the brain, as we know, requires a lot of energy to stay active. You understand? It requires about 20% of the body glucose. Very, very essential. So moving on, what is this glycogenolysis about? The process of glycogenolysis involves the breakdown of glycogen, as we have said already. You can see glycogen is being broken down to glucose. This is just a little demonstration of what it's about. Then, what are the steps now? Very, very important. Consider this. I'm just trying to make it as simplified as possible. The steps of glycogenolysis, from the term glycogen, it is first of all acted upon by the enzyme glycogen phosphorylase, which is the rate limiting enzyme. Please, very important, rate limiting enzyme. Why is it rate limiting? Because it carries out the longest step. You understand, in terms of duration, it takes its time to break down the glycogen. Then the debranching enzyme, now on the other hand, this is very important too, because glycogen, the structure of glycogen, it is a branched polysaccharide. Very, very important. You understand? Very, very essential. It's a branched polysaccharide. You understand? Means that it has two types of bonds present. It has the alpha 1 4 glycosidic bond and also alpha 1 6 glycosidic bond. Very, very essential. If you have a note or paper, keep jotting down what I've been saying. You understand? So this will lead to the formation of what is called glucose 1 phosphate, then which is acted upon by another enzyme called phosphoglucomutase, which is going to form glucose 6 phosphates, and glucose 6 phosphates then converts to form glucose. Very, very important. So as you can see here, the muscle, the liver, means that the muscle, the important parts of the body that helps in this process of breakdown of glycogen are two. They are muscle, the muscle and the what, and the liver. The muscle is a stingy organ because it does not send out the glucose, rather it uses the glucose. When the liver, on the other hand, is an important organ because it's very generous. It will, when it has broken its its glycogen, it can use part of it and also send most of it out for the body to maintain blood glucose and for other organs to use up glucose. Very, very essential. Moving on, the glycogenolysis, to make it further clear, you can see this image now. It actually depicts how this whole process of glycogenolysis happens. You can see now, look at it. These are the glucose molecules, very, very important, and the bonds are also there to indicate it there. The ones here are the alpha 1 4 glycosidic bond or linkages, while the other one that causes the branch there is called the alpha 1 6 glycosidic bond or linkage. So when acted upon by the first enzyme, which is the rate limiting enzyme, I said it before, now be the rate limiting enzyme, the glycogen phosphorylase breaks it down to form, each of them forms the glucose 1 phosphate molecule. Is very, very important. It does it to a point, because this is the non-reducing end. Very, very important. That's what it acts on. Very, very important. Breaks them down to form the glucose 1 phosphate molecules. Then it gets to a point when it is about 4 molecules to the branch point to stop. That guy cannot work again. Then another guy will come into the picture, which is the debranching enzyme. It has two activities. It has a transferase activity. It also has a glucosidase activity. From meaning transferase, it transfer, or it transfers. Then glucosidase on the other hand means it breaks down glucose. You understand? So the one does this guy now, transferase activity means that it will transfer the three of them, these three glucose molecules now, or three linked glucose molecules, yes, to the other side, the other branch, or the other part, you understand, of the glycogen. Then the glucose remaining at that point now is also broken down again, you understand, so to form glucose. Then the remaining parts now, the remaining glycogen is no longer branched, so it is left for the phosphorylase now to continue its activity, to keep breaking down. That's why it is regarded as a rate limiting enzyme, you understand, because its activity is quite essential for the process of glycogenolysis. So what I talk about is now, say this glycogen now gets branch, and they get two type of bonds. Get alpha 1 4 glycosidic bond. You also get alpha 1 6 glycosidic bond. The first guy, which is the rate limiting enzyme, means say the activity, the time they happen longest, then they take time to happen. You go doing work, will reach one point. So when you reach about 4 glucose molecules to the branch point here now, you go carry the three of them, go the other side. You understand? Then the other activity we get now, glucosidase activity, alpha 1 6 glucosidase activity, means say if they break alpha 1 6 glycosidic bond, will come from glucose. Then the other pata will come continue with it they do normally, which is glycogen phosphorylase. That's it. So like I do always, I must always give a question to actually buttress what I have been teaching so far. So a 6-month-old infant presents with severe hypoglycemia, hepatomegaly, lactic acidosis, and hyperuricemia. A glucagon challenge test shows no increase in blood glucose. Which enzyme is most likely deficient? Is it glycogen phosphorylase? Is it the branching enzyme? Is it glucose 6 phosphatase? Or muscle phosphorylase kinase? So take your time to make your research and check from what I've taught so far. What do you think the answer should be to this question? Then send your comments, put in your comment section, and ask any question you have that you think is necessary to the topic in question. So thank you very much. Don't forget to like, don't forget to subscribe, don't forget to share to your colleagues to help them learn this process too. Thank you very much. I appreciate it.